Okay, good morning, everybody. So today we have an extraordinary guest, Professor Manuel Pombal from University of Vigo in Spain. This is it seems this university is the most to the west in European continent. University, and not only the most Western university, but also with a very pleasant temperatures and weather, with magnificent views of Atlantic Ocean. So the Vigo University is a similar size of our university, but our special guest, he did the PhD also in Spain, in Santiago de Compostela, and after several adventures, he settled around Vigo. And he is working at the Vigo University. However, in his curriculum, there are many interactions, collaborations with Karolinska Institute in Stockholm. So he is very close to Nobel Prize winners, I would say. We like to be close to the Nobel Prize, so we like you more than. Okay, but he's a neurobiologist, and his preferred model of research, I don't know whether it's also the preferred model of teaching, but the preferred model of research, this is a fish nervous system. Maybe he will show us some pictures of his fishes. And but for scientists, the most important thing is the list of publication, and this list of publication is very long. There are maybe even more. 58 research papers in a very good journals like Neuroscience, European Journal of Neurology, and Frontiers in neuroanatomy, and in Frontiers in Cell Development, and so on. So these are very good journals for neurobiologists. And in addition to this, there are some book chapters and also educational publications. And he's also experienced in doctoral thesis supervision. He was a main supervisor of 3 PhDs and a co-supervisor of 4, according to your CV. So it's a very good teacher and scientist, and it will be a pleasure for us to hear the first lecture about, as you can see, stem cells and tissue engineering. So, Professor Pombal, the floor is yours, and we are eager to hear you.
Thank you. So, thank you very much for your presentation. Okay, so today I will talk to you about stem cells and tissue engineering. So this is the summary of my talk. First, I will introduce tissue engineering and regenerative therapy, and then we will move to the source of cells, which are going to be the stem cells, and I will comment on the characteristics and utility of the stem cells, niches, different types of stem cells. And then we will have a comment about some specific steps for tissue engineering, like the isolation and proliferation of the cells we need, some signaling and scaffolding that is also needed for the preparation of the specific tissues. And then I will comment about different specific problems we will find in all the procedures, to end with a few notes on some recent advance in this field. So, tissue engineering use stem cells and biocompatible materials to produce in vitro structures similar to normal tissues, mainly with the aim of being implanted in an organism to repair damage due to pathologies or accidents. On other occasions, they are used for research, such for testing environmental conditions, drugs, personalized medicine, or simply to do basic science. Regenerative therapy attempts to repair tissues and organs in situ. In both cases, they are multidisciplinary sciences where different types of professionals, such as biologists, doctors, engineers, physicians, chemists, and so on, participate. Tissue engineering and regenerative therapy are also extremely complex disciplines that require a deep understanding of tissue development and homeostasis, as well as of cellular differentiation processes. There is no general protocol to generate tissues, but rather the methodology must be adapted according to the tissue we are working on. Thus, what works well for poorly vascularized tissues with low metabolic rates, such as skin or cartilage, doesn't work well for other types of much more complex tissues. On the other hand, knowledge of materials, design and composition, and their interaction with cells is very important. In 1977, the vacant mouse had a great social impact because it had a human ear grown on its back. Despite this and other advances in tissue engineering and regenerative therapy since then, there are not many treatments approved by state agencies that are being widely used in hospitals, although the number of clinical trials grows year after year. So there are several key elements we need in tissue culture, such as a source of cells, a supporting structure on which to grow the cells, and a chemical environment suitable for cell differentiation, as we are going to see in next slides. To grow or repair any tissue, we need cells to form it. Actually, we need a high number of cells of different types. Of course, those cell types that are part of the needed tissue. And they must be differentiated. We could take cells from other parts of the body and replace damaged tissue. Which is known as a self-transplantation. But this is not possible in many cases. For example, if you have a small burn, you can take small pieces of skin from other places on the body and seal the burn. But if it is a large burn, it is not longer possible. The same thing happens with other tissues, such as cartilage, bones, or entire organs. The body's tissues are constantly renewed, as you know, under normal conditions. That the cells die and are constantly replaced. In some situations, if there are burns or pathologies, the renewal rate is accelerated. Every day, millions of red blood cells disappear and are replaced by bone marrow cells. All red blood cells are replaced every 6 weeks. The entire skeleton is renewed every 6 to 8 years, and half of the cardiomyocytes are renewed at 50 years. All this capacity to produce new cells is based on one specific cell type, stem cells. Stem cells are undifferentiated cells with the capacity for proliferation and differentiation that give rise to all functional cells in tissues. There are different types of stem cells, but they all share two properties. One is self-renewal, okay, so that they proliferate and make identical copies of themselves, and this proliferation occurs in a specific place in the embryo of adult organism called niches, where stem cells are controlled. When stem cells divide, some of the cell divisions are asymmetric. So, in this case, one of the resulting cells begins to differentiate. This cell first become a progenitor cell or transit amplifying cell, which will divide many times to increase the number of cells in the population. At a certain point, all the cells in this population will differentiate. The asymmetric division can occur due to external or intracellular signals. Stem cells are located in very precise and characteristic places in the tissues called niches. In these niches is where they proliferate under control conditions. These niches are made up of cells, extracellular matrix, or a mixture of both. Niches are therefore some kind of microenvironments where adult stem cells live. By emitting paracrine or juxtacrine signals, cell-to-cell or cell-extracellular matrix adhesion, the niches control the proliferation and differentiation of stem cells. The composition and organization of the niches is different depending on the type of tissue or organ. This drawing shows the cellular composition of the adult ventricular, subventricular niche and domains of B1 cells in the central nervous system, which correspond to the neural stem cells. So this is the B1 cell, which is the neural stem cell, which are located in a few locations in the brain, as we will say later on. So these B1 cells are surrounded by many multiciliated ventricular cells. B1 cells give rise to intermediate progenitor cells, the transit amplified progenitor cells, which are the C cells there in green color, that divide to generate the neuroblasts, A cells in red in the drawing. The neural stem cells retain epithelial properties with an apical process containing a primary cilium there that contacts the lateral ventricle and a long basal processes that end in blood vessels. Therefore, B1 cells can be subdivided into three domains. We have domain 1, proximal or apical domain, that contains the primary epithelium and is in direct contact with the CSF, the cerebrospinal fluid, which within this domain, cells can interact with soluble factors within the CSF and signaling molecules from neighboring ependymal cells. The domain 2, or intermediate domain, in medium blue color there, is in close proximity to C cells, neuroblasts, and neuronal terminals and other supporting cells. Therefore, different cell-to-cell interactions between B1 cells and their progeny will occur within this compartment. Domain 3, or distal domain, in light blue color, comprises basal processes ending in a specialized end foot that contacts blood vessels. So blood-borne factors and endothelial derived factors may act on B1 cells in this domain. In the other brain, there are two specific places where we can find stem cells. One is in the one is in the hippocampus at the level of the dentate gyrus, and the other is at the subventricular or subependylar zone, close to the lateral ventricle, where a rostral migratory stream is originated towards the olfactory bulb. In the bottom panels, we can see the schematic drawings of the cell organization of the niches of these places. This is another example of another niche, in this case to renew the cells of our skin, so that the basal layer we can find the stem cells, epidermal stem cells, and the transient or progenitor amplifying cells, which are close to the basement membrane. So these stem cells will proliferate from time to time, will divide from time to time, and originate these transient amplifying cells, which are going to divide many times to produce cells. And when there is an asymmetric division, one of the cells will start the process of differentiation to produce the, let's say, other somatic cells. So moving away from the basement membrane and passing through the different layers, spinous, granular, and to finally end at the level of the stratum corneum, where they will finally be released by the squamation. This is another example of a niche, in this case at the level of the intestinal glands. So the cycle here begins at the bottom of the glands, where we will find the CBC cells, okay? And we'll end at the upper part of the gland with the different types of differentiated cells. So in the journal, is it possible to distinguish how is the tissue renewal processes? At the bottom, we will find the CB cells, which are in green in the scheme. And then we will find the transient amplifying component, which is blue here, where these progenitor cells proliferate, and then they will differentiate into different types of cells. In this 3 image, we can see the expression of Ki67, which is a protein that is located in the nucleus and is a marker of cell proliferation. So if we have a look, we can see as at the bottom there is no expression where the stem cells are located, but we can see that the level of the staining corresponds with the transient amplifying component, where cells are proliferating. And in the upper portion, where are the differentiated cells, there is also no expression. In this other image, we can see that most of the cells express the marker T67. And this corresponds to an image of an epithelial tissue of a colorectal cancer, which means that most of the cells are proliferating there. Now I will comment a little bit on the different types of stem cells. Apart from the totipotent stem cells, there are two main types of stem cells according to their differentiation capacity: pluripotent and multipotent stem cells. Pluripotent stem cells are stem cells that can differentiate into any cell type in the body. Only the zygote and the first blastomeres are totipotent. That is, they can give rise all the cells in the body, but also cells of the extraembryonic structures. Embryonic stem cells are one type of pluripotent cells that are obtained from the blastocyst stage embryo of day 5 in humans, quite early in development. And these embryonic stem cells are known as cells of the inner mass. And we can see there this group of cells inside the blastocyst, which correspond to this inner cell mass. These embryos are donated by their parents to science. And they are, of course, also obtained from the blastocysts of other mammals for preliminary studies. Embryonic stem cells give rise to cell masses in a short time, and their differentiation capacity makes them very versatile. However, several problems still need to be solved to use embryonic stem cells in the clinic. Working with these cells is very expensive in time and money. The culture media are complex and normally obtained from animals, which leads to problems such as prions or the incorporation of antigens into the cells themselves. However, the main problem in clinical practice is ethical, due to the use of human embryos. In addition, they produce immune rejection, like any other transplant. Induced stem cells are another type of pluripotent stem cells that are generated from differentiated cells by the differentiation processes, by change in gene expressions. Induced stem cells can be obtained from different human cells, modifying the expression of only 4 genes: OCT4, SOX2, c-MYC, and KLF4. Which are known as the Yamanaka genes. Actually, Yamanaka got the Nobel Prize in 2012. Nowadays, some other genes are also used, like nano, Lin28, some others. There are many somatic cells that can be used as the source of these induced stem cells, and there are different strategies to reprogram these cells. For example, injection of mRNA of these 4 genes can produce induced stem cells in 2 to 4 weeks. Then they have to be cultured to generate a high number of pluripotent cells. However, these cultures have to be cleaned or filtered by using flow cytometry, since they are relatively heterogeneous. This type of cells, you can see at the bottom of the drawing, can be used to study some disease, to identify new biomarkers for improved diagnostic procedures, or to develop new drugs, or for cell replacement therapies. Multipotent cells, the other type of stem cells, are those that are found in adult tissues that have the capacity to proliferate and give progenitor cells, which will differentiate into the typical cells of that tissue. Adult stem cells are multipotent cells found in all tissues with the capacity for repair. There are different types, such hematopoietic, epidermic, nerve, cardiac, mesenchymal, and so on. A characteristic of adult stem cells is that they are heterogeneous within the organism. That is, their characteristics depend on the tissue where they are obtained. By differentiation, they give rise to the typical cells of the tissue where they are found. Although there are examples of transdifferentiation. For example, a hematopoietic cell can give fibroblasts or neurons. A special type of multipotent cell that appears in many tissues are the mesenchymal cells, which are derived from the embryonic mesoderm. They are the most studied and clinical use adult stem cells. They are found in the umbilical cord, placenta, adipose tissue, bone marrow, dental pulp, blood, and other tissues. They can also be obtained from embryonic or induced stem cells. They are very versatile and easy to obtain, and the differentiation potential is enormous when treated with the right signals. They can differentiate into neurons, adipocytes, myoblasts, osteoblasts, etc. They are very usually isolated from tissues within flow cytometry. One of the most important characteristics of mesenchymal cells is the great diversity of molecules they can release, which is known as the secretome. The secretome, actually, the secretome of these cells is truly spectacular. These releasing molecules prevent inflammation, include trophic factors, allow communication with the immune system, and regulate its response. They also stimulate angiogenesis. In this way, they establish optimal conditions in the tissue for tissue regeneration. Some therapies include mesenchymal cells in conjunction with allogenic tissues or organ transplants due to their ability to modulate the immune response. Another important characteristic is that they can migrate to damaged places so that they can be injected in the blood system. They are, however, heterogeneous. Since depending on their origin, they express different genes and have different potentials. So that means that the appropriate types of mesenchymal cells must be chosen according to needs. In 2021, there were around 1,000 clinical trials underway using these cells. Of all the clinical trials with mesenchymal cells, only a few are in phase 3, and they have to do with tissue regeneration. For example, mesenchymal cells have been injected to cure Crohn's disease and retinitis pigmentosa. Hematopoietic stem cells are another highly studied type of multipotent cells. They are the precursors of all blood cells, including those of the immune system. Bone marrow transplants have been done for a long time, and a transplant with hematopoietic stem cells can completely regenerate the blood and immune system. They are, however, difficult to grow and to obtain in high numbers. Nowadays, these cells are extracted from blood after stimulation with trophic factors. The placenta and umbilical cord are two other important sources for this type of cell. In the case of the bone marrow, there are somewhat diffuse niches for stem cells of different types. For example, the niche of hematopoietic cells is divided into two physical spaces: the bone surface and the perivascular region inside the bone marrow. Even the molecular environment of the bone marrow is often considered to be itself a part of the niche. since there is a cocktail of molecules generated by other cells in the bone marrow, including osteoblasts and mesenchymal stromal cells, as well as the endothelial cells themselves. So once we have the cells, we need to isolate and do the proliferation. As already commented, any of the types of stem cells choosing as a cell source need to be first isolated and then cultured to increase their number. The most common isolation procedure consists of using antibodies against surface antigens that identify the different types of stem cells and then separate these stem cells from the rest of the cells using flow cytometry. The power of this technique is really high, as about 10,000 cells can be analyzed per minute. After isolation, the stem cells are cultured in media that favor their proliferation and undifferentiated states. However, stem cells are very sensitive to environmental change. And in the same culture, cells in the periphery can respond differently to the differentiation factors from those in the center. This implies that generally after culture, to increase the number of cells, a new selection must be made to discard those that have changed their phenotypes. The other important thing is this signal molecules or signals and the scaffolding needed to create the tissue or the organ. So once we have a large number of homogeneous stem cells, we must differentiate them into the cell types we need. In normal tissues, during embryonic development or in pathological situations, the stem cells develop in a certain environment that guides them during the differentiation processes. This implies that they have to receive specific signals in a certain concentration, in a specific sequence, and at an appropriate time. So this is not only for differentiation, but also to achieve the final cell number and a suitable position of each cell type in the tissue. There is a multitude of signals depending on the cell type and physiological state. Cells do not respond to all signals, although they do respond to a larger number and to many of them simultaneously. Signals can be molecules anchored to the extracellular matrix or to other cells, or be soluble molecules. There are other types of signals that must be taken into account: the three-dimensional organization, the mechanical properties of the adhesive characteristics of the framework, where they will be attached to grow and differentiate. In normal tissues, this environment is created by the extracellular matrix and adjacent cells. To survive, most cells must be attached. This adhesion is not only for holding or crawling, but also for receiving and sending information. There are many adhesion molecules that simultaneously act as receptors: integrins, cadherins, immunoglobulins, selectins, proteoglycans, CD44, etc. Cadherins bind cells together, important for creating and maintaining cell populations together. CD44 receptors are important for adhesion to carbohydrates such as hyaluronic acid, while integrins recognize protein elements of the extracellular matrix and are also very important for adhere to synthetic elements. All of these adhesion molecules are able of adhering and triggering signaling pathways at the same time. Here we have an integrin which is binding different molecules at the level of the extracellular matrix and at the level of the cytoplasm, can bind either one of the elements of the cytoskeleton, actin filaments, or other type of molecules, so that apart from the adhesion properties of these molecules, they can also participate in different types of signaling pathways, like inside out, mediated by these cytoskeleton actin filaments, or outside in signaling through other molecules that are going to start the signaling pathway at the level of the cytoplasm. Another level of regulation of the cell's modification is the activation and inactivation of integrins. So the adhesiveness is an active process controlled by the cell itself. As we have said previously, integrins also function as receptors, in this case as mechanoreceptors. And the transduction of adhesion is called mechanotransduction. So the interaction between cells and extracellular matrices is what engineers must understand when synthesizing new materials to manufacture synthetic matrices. Many molecules important for cell differentiation are soluble molecules or are anchored to the extracellular matrix and are released during the remodeling. These molecules bind to traditional receptors and trigger an intracellular pathway. Among the most abundant and most diverse receptors in cells are those associated to G proteins, GCPR receptors. These receptors are characterized because their activation modifies the G proteins that initiate the signaling pathway. Other very abundant receptors are the tyrosine kinase receptors, which are associated with enzymatic activities, mainly kinases. The reconstruction of tissues, either in vitro or in the body itself, involves the use of a structure or a scaffolding to which the cells adhere. These scaffolds can be natural, synthetic, or a mixture of both. Within the natural scaffolds are the extracellular matrices themselves. These are obtained from extracting identical tissues, usually from animals, and subsequently eliminating the cells they contain through a process called decellularization. These processes should not affect the molecules in the matrix. Its result is measured by the amount of DNA that should be 0, that means no cells there, or very minimal. The decellularization can be achieved with physical treatments, including sonication, shaking, or freezing. There are also some enzymatic treatments, and in some cases, detergents are used. The procedure used depends on the tissue. After decellularization, the matrix must be sterilized before implantation or use in vitro. For this, high temperature cannot be used because it denatures the molecules. But radiation, ionization, gases, etc. can be used. These cell-free extracellular matrices have already been used clinically. There are currently more than 40 commercial products. More than 3 million patients have received implants with these scaffolds, mostly of porcine origin. These matrices can be implanted directly into damaged areas. In this slide, several applications of cell-derived extracellular matrices in basic research are shown. Synthetic matrices have advantage and disadvantage. One advantage is that they can be produced on an industrial scale once optimized. Other advantage is that their components, 3D organization, and properties can be precisely controlled. Once standard, these processes are cheaper to obtain and can be produced in large quantities. They must be made of biocompatible materials and, in addition, be degradable. The latter is very important for adequate tissue regeneration. Among the most used synthetic matrices are hydrogels, which are highly hydrated synthetic or natural polymers, and they have high porosity. And in addition, they are soft and deformable. Mixed matrices also can be generated by using molecules such as collagen, glycosaminoglycans, and other disaggregated from tissue matrices and assembled in the laboratory, either among themselves or mixed with other components. Mixed matrices can also be constructed sequentially. First, the scaffold is made with biomaterials. The scaffold then is seeded with cells. The cells reconstruct the extracellular matrix by culturing it in a bioreactor while eliminating the initial material. The cells are subsequently eliminated and the decellularized matrix scaffold is implanted. This has been done for blood vessels, for example. In this case, the scaffold will be repopulated in situ by the patient cells. Regardless of the material used, the matrices must have a series of properties that affect the tissue to be grown and that vary depending on the tissue. Getting all of them is complicated, and they are the main disadvantage of this type of scaffolding. Some of the most important properties are they must be biocompatible. That is, they must be accepted by cells and they should not produce cellular damage or alterations due to their composition. Adhesion is also essential for undifferentiated cells to proliferate. This adhesion should not be very strong or very weak, since it can affect proliferation and differentiation processes. It is interesting that the matrix itself contains survival and differentiation factors integrated into its own structure. It is even more interesting if they can be released progressively and over a certain time. And even more interesting if there are microregions with different chemical characteristics that can affect the cell differentiation processes differentially. The spatial organization of its molecules, such as porosity, frameworks, etc., is another important factor for cell proliferation and differentiation, which depends on the tissue considered for repair. In addition, microdomains or heterogeneity on scales of the order of micrometers may be needed, both in 3D organization and in distribution of the type of molecules. For example, hydrogels lack this property. These microdomains also affect the proliferation and the differentiation. Another factor to take into account is the mechanical properties, which include rigidity, elasticity, or hydration, which determine the behavior of cell types. For example, a certain elasticity of the matrix is necessary for correct differentiation of muscle cells from mesenchymal cells. The matrices have to be degraded and replaced by others synthesized by the cells themselves. Actually, the degradation process itself involves the release of molecules that facilitate tissue repair. As mentioned before, although stem cell therapy has been researched for many years, and although there are numerous trials in the experimental phases and some in clinical trials, there are very few therapies based on tissue regeneration or tissue engineering approved by national health agencies. And this is because there are still general problems and some specific ones to solve. It must be taken into account that the procedure must be standardized and the conditions of isolation, culture, differentiation, etc. must be fully controlled, must be reproducible, and not produce harm to the patient. One of the bottlenecks in bringing these treatments to the clinic is obtaining a high number of cells in vitro, typically tens to hundreds of millions per treatment. In addition, the cells must be cultured under very specific conditions that comply with sanitary standards and that shows minimal variations between batches. The cultivation system must be scalable to an industrial level. Matrix properties such as microdomains, as I already commented. An additional problem to the heterogeneity of the cell populations obtaining is the heterogeneity of the cell populations obtained in stem cell cultures. That is, apart from the stem cells we want, other unwanted cell types will also appear. Therefore, the specific cell type we need must be selected by using flow cytometry. Once we have a good number of cells, we need to differentiate them, avoiding poorly differentiated or undifferentiated cells, and cells that have undesired phenotypes. This can in part be controlled by the properties of the matrices, natural or artificial. There are clear examples during the differentiation of mesenchymal cells where rigid matrices led to differentiation into bone cells, while more relaxed ones led to muscular phenotypes. This has also been seen with artificial matrices. If the stem cells are injected directly into the tissue, many cells die from mechanical damage or do not adapt to the new environment. For example, hydrogels embedded in growth factors have improved survival and adaptation. They may even have anti-inflammatory factors that decrease the immune response, which increase graft viability. If tissues fail to create blood vessels during tissue regeneration or after grafting, the innermost cells will die. To solve this problem, there are two strategies. In the first one, the appropriate growth factors and cells are added to promote vascularization. In the second one, the vascular system is built in vitro around which the tissue-specific cells grow, and everything is subsequently transplanted. Numerous experiments in mice and other mammals have shown that it is very difficult to avoid undifferentiated cells in a stem cell culture, even after differentiation processes. If these cells are injected, they can create tumors such as teratomas or teratocarcinomas. All cells that are not derived from the patient themselves, I mean allogenic cells, can produce immune rejection. So the most suitable will be induced stem cells obtained from the patient himself. However, experiments in mice have shown that these cells can also generate antigens during the process of their differentiation and in vitro culture. Concerning animal models, most engineering experiments have been carried out in laboratory animal models. Although they are a good tool to know the general steps of the processes, they are not completely transferable to humans. This implies that the entire processes must be reconstructed to work in humans. Finally, working with embryonic stem cells means using human embryos, which raise ethical problems. One branch of tissue engineering seeks to use these technologies not to repair a tissue, but to study that tissue outside of the individual. For example, organoid cultures can be created and try to recreate the real organs as close as possible. In these structures, drugs can be tested in a general way or to see the effectiveness of drugs in a specific patient, known as personalized medicine, or study the response to any type of stimulus in a high control environment. Cultures called biochips have also been developed. They are cultures of cells encapsulated in devices that are subjected to different conditions to study the responses. Other type of cultures called organoids are obtained from stem cells. Organoids of the brain, retina, and digestive system have already been obtained from induced stem cells in the laboratory, but still without vascularization. The term biomanufacturing consists of the creation of 3D structures similar in organization and composition to natural ones, and with the appropriate cell types. One of the biomanufacturing techniques is bioprinting, which use computers to transfer components that create a pattern and assembly of cellular and artificial molecules into a larger organ-like structure. In this slide, there are different types of 3D bioprinters. As you can see, there are many different types of these bioprintings. And just to finish, I will comment a little bit on some artificial tissues that are already used as in the clinics. Okay, and here there are some results from the tissue engineering group of the University of Granada in the south of Spain. They have just developed different types of artificial tissues. And you can see also the chronological time they spend from the first design and the point the first one was implanted in patients. For example, they did the first design of the first artificial cornea in 2006, and the first implantation in blind patients was in 2014, okay? 8 years later. The first design of artificial skin was in 2012, and the first implantation in large burns patients was in 2016. And in the case of the palate, the first design was in 2016, and the first implantation was in 2024. And they are also designing artificial nerves. So these are some of the great designs and applications from tissue engineering. And I really thought this type of research is quite promising for the next decades. That's all. Okay.
Thank you very much, Professor Pombal. And this is really very exciting and futuristic. But also, as you've shown, many problems to solve is the work for you to discover new techniques and new solutions to make it more maybe not realistic, because it is realistic, because there are several therapies, but to solve many problems, to use it in many more cases. Do you have any questions? So you are welcome to ask. Marek.
Oh. I have a quite loud voice. I see. Very interesting topic, and very difficult topic actually. And I have a short question. What about onco stem cells, which develop some tumors?
Yeah, well, there are many possibilities. I commented on some of them because, actually there is a specific niche also for these onco stem cells, which then in this kind of niche, they are not really, their proliferation is not under control. So that means that they grow and then can freely somehow migrate to different final destinations. And the point is that this the differentiation processes of the stem cells is so complicated that in some cases there are some, let's say, mutations that can originate this kind of tumor cells. But actually, as I'm not a specialist in this field, I cannot go any further. Yeah. Sorry.
Thank you. More questions? So I may ask my own question. So what's your opinion for using these stem cells in gene therapy? Whether we should develop treatments to treat the adult differentiated cells or the stem cells?
Well, you have somehow advantage and disadvantage. It's difficult, and it depends on the specific situation. For example, if you induce stem cells, one of the important things is that you are using the cells of the patient. So that means that you will avoid immune rejection. And that is somehow really important, okay? Because it's one of the major problems when we do a graft or an implant. So from that point of view, I think that this kind of personalized medicine is going to be important in the future. But I cannot preview what will be more important, because if you can use stem cells that you can implant to different type of patients, then that will be great, because with let's say one simple procedure, you will get tissues for different type of patients. If that is the case, probably that will be much better than to use the cells from the patient himself. But maybe that for some specific tissues or organs, this is more difficult to do in a general way, and then in that case, the best solution will be to use induce stem cells obtained from the patient himself. So I think that there will be different types of situations, and which one is going to be more effective, I will not be able to pronounce myself.
Okay, thank you. And maybe also very important, but the argument is, what is the final price of the therapy?
Yeah, of course. Yeah, that is so important.
Yeah.
Because as you see, these procedures are really extremely long processes, and there are many different steps where we can somehow get problems. So, and to solve all of them is not so easy. So that means that to get this standard, these procedures, is one of the major tricky points. And of course, once you have everything standard, it will be easier. I mean, yeah, but it's a long way, actually.
Thank you very much. We have more questions. Agnieszka, later Ewelina.
Okay. Thank you for a nice talk. I have a question about using stem cells for producing the blood cells. Is it possible? Because we all know that there is a lacking of the blood in the blood banks. So
Well,
do you think we can use it?
Yeah, actually, I cannot answer the question because I have no, I mean, sadly, I don't exactly know if this kind of therapy is now in clinics, okay? But of course, as I said, there are many research in relation to the bone marrow, okay, application, and I think there are already some therapies on the clinics, but I don't know exactly how they work or how many, you know, how effective they are. But
Thank you. Ewelina?
Yeah, I have a question because we have our scaffold and we want to use it to produce, for example, organs. And we can have also tumor because we don't know how it really differentiate. And my question is, can we somehow prevent inside of the scaffold the tumor differentiation? Because we know somehow how the tumor niche works and the organs, the normal organs works. So maybe if we put something in the scaffold, which could prevent the tumor differentiation, the organs will be less, we have less chance to get tumor, not the right organs.
well, let's see if I understood well. The point is that, of course, with all this preparation of different organs, like heart, there is some kind of video on the internet where they use a pig heart and they do the decellularization process, so you get only the extracellular matrix, and then you can seed it with the stem cells. So, of course, you will run these experiments in vitro and see how the organ develops, how the stem cells proliferate and are able to produce the 3D structure of the organ with the different cell types in the different in the right place. I think, there are also you can control somehow better the let's say the production of tumor cells when you are growing the stem cell population. If you must select the cells that you are going to use to create your tissue or your organ. The point is that I don't know how high is the possibility to create or to generate tumor cells during the process of the building of the tissue or the organ himself. Okay, so I suppose the possibility of getting tumor cells is higher during the culture of the stem cells than when you add these stem cells to the scaffolding to grow the organ. But in reality, I don't know the numbers or the possibility in either comparing either phases of the procedure.
Thank you.
Thank you. Other more questions? Okay, so thank you very much for your lecture. It was inspiring and interesting, and so we close this seminar. Thank you.
Thank you.